achieving this regulation. So here's PFK, aldolase, and triose phosphate isomerase, which are 3 reactions in a row in glycolysis, right? So, as we said, aldolase and triose phosphate isomerase, that is to say enzymes 2 and 3, are not regulated. And because they're not regulated, they're also fast. There's nothing on there to slow them down, right? But also because Q equals K for those 2 enzymes, they're fast the other way as well, right? It's because they're good enzymes. And there's no inhibitors around to stop them. They're not regulated in any particular way, right? So they're at equilibrium, and they're happy to do whatever PFK tells them to do, as far as this example goes. Now in practice, they're also happy to do what the combination of PFK and hexokinase and pyruvate kinase tell them to do, right? But we're just going to settle for looking at what happens here. So PFK is the regulated enzyme, and for which Q is far from K. Q is 0.03, K is 315. So the driving force this way is such as to produce a velocity that's on the order of, so these numbers are going to be a little bit fudgy, but it'll be on the order of 1,000-fold faster this way than this way. Right? And it might be a little bit more or less than that. But it's not important for the example. The point is that it's far from equilibrium, right? And it wants to go this way. So the net rate is just 10 going forward. So this, because there's always a lot more B than A, this guy will be in excess of C. So this will go forward by how much? By a distinction that is provided by how much B this can ever make, right? Which is 10, and then the same for the next one. So why is it that this number is 200 and this number is 500 and this number is a lowly 10? Because that's part of what the allosteric inhibitor does, right? It's an allosteric effector, but the effect of it is to actually turn down the activity quite a lot, right? So what we have is a combination of things. We have the allosteric effector on PFK, which we will learn about as far as what it is and how it works next time. Operating to slow everything down. And then we have the combined effect of regulation in the cell that's generating metabolite concentrations that are such that Q is far away, far, far lower than K, meaning that A is very, very low in concentration. Sorry, A is very, very high in concentration and B is very, very low. So it's going to go this way, right? So if it weren't also turned down a lot, and that driving force was so strong, and these guys aren't going to do anything about anything, right? Then all the A would just shoot over to B, right? If the enzyme were really operating as well as it could. The A would shoot over to B, and then you could shoot right through. And then there wouldn't be much A left. And you could say, well, okay, that's the way it should go. But you know, what is A for pyruvate kinase?
Glucose. Glucose?
Glucose?
Pyruvate.
Sorry. Phosphofructokinase. What is A? Phosphofructose kinase, the third step in glycolysis, is binding.
Fructose 6 phosphate.
What?
Fructose 6 phosphate.
Okay, but what else? So, ATP, right? And what happens if all the ATP turns into some phosphorylated sugar? Now your ATP concentration is very low. And you have just violated the cardinal rule of all metabolic regulation, like the #1 thing that all cells have to do, in addition to replicating their genetic material, which is to keep their ATP high, right? So these principles are applied to pretty much every ATP metabolizing enzyme in the cell, or the vast majority of them. Right? If not, you would lose your ATP. Right? You would suck up your thermodynamic driving force, right, by not having your ATP concentration high. And then your Q, well, if your ATP concentration were not high, then your Q would be high. And then your Q would be like approaching your K. And then you wouldn't regulate this guy anymore. Right? So that would mean there'd be no way to turn this guy off. Well, that would mean you'd eat up all your fuel even when you weren't supposed to. Right? Does this make sense? So the text on this point, it's accurate, I find. I think it's well written, but it's, you know, it's a little dense. It bears repeated reading. But you're kind of at the core of how biochemical thermodynamics really works in the cell, if you kind of appreciate these points. Yeah?
So would you say that speed is a trade-off for regulation?
Yeah, the overall pathway is not going to go, but it doesn't need to. And that's another aspect of all the balancing in the cell. So the pathways don't need to go this fast. The enzymes have the capacity to go a lot faster than they actually need to, as far as the physiology of the cell says. Right. Suppress the activity of enzyme 1, limit conversion of A to B, maintain Q low. Questions? Okay.
So,
What else?
I have a
Yes, sir.
So is it then typical, so the parts that require ATP be regulated to prevent that from happening where you just overuse your ATP?
Right. Yeah, that's the most important principle. Yeah. Now it applies to other metabolites as well. It applies in a pretty significant way to things like NADH, because the cell, it's also very important to maintain the redox balances. And we talked about how the NAD to NADH ratio in the mitochondria is low, and the NADPH to NADP ratio in the cytoplasm is high, which is a consequence of the different metabolic specializations of those compartments. Right? But cells also have to make sure that those concentrations stay that way, and that's going to be done by regulation as well. Perhaps slightly less critical than the ATP. Okay. Let me go past this for a second. So here are some typical concentrations of ATP. It's also 5 or 10 millimolar, something like that, is a typical ATP concentration in the cell. If there's a lot of ATP being consumed for whatever metabolic reason, and the concentration is high, the relative change is not nearly as high as if you had a reaction, for example, where you changed the AMP concentration. So ATP often goes to ADP, but then there's another enzyme around, there's a name I can't quite remember, it's in your book, that converts two ADPs to an ATP and an AMP. Anybody remember the name of that enzyme?
Adenylate kinase?
I think it might be adenylate kinase, yes. I'll check on that. I think it's adenylate kinase.
Is it one of the G proteins?
No, it's not a G protein. It's a metabolic enzyme that works with these different levels of the adenosine nucleotides. It's not the NDP kinases that shift around ADP and GDP. It's a different enzyme. But whatever the name of it is, and I think that's right. ATP goes to ADP if it's being consumed. That's much more typical to go to ADP. There are reactions that go ATP to AMP and produce pyrophosphate. That has the advantage of pyrophosphatase thermodynamically driving it. But if you have another enzyme that takes ADP and makes ATP and AMP out of it, then that's the same idea. So you deplete. So the action of those enzymes would result in a huge elevation in the AMP concentration, a relative change of AMP, which is very large. And so that can be exploited